My work is also partially involved in the de.NBI cloud. And today I will be presenting the Bioinformatics Tools that is built based on the Apache Spark framework. I'll give you a little bit background here. So the motivation of my work is to address the big data challenge in life science, particularly the NGS data sequenced from the next generation sequencing. As we all know, the human genome all the genomes are extremely large. The humans, for instance, has 3 gigabits of nucleotides. And for modern technology, we are not able to sequence the entire chromosome in one continuous sequence, because they are extremely small and fragile. So what the sequence do is instead of sequence the entire nucleotides, they fragment the nucleotides into fragments, small pieces, and sequence them in a redundant way. And what you get is a piece of nucleotide sequences that is overlapped. And for the bioinformaticians, their work is to use this kind of overlap to reconstruct the genomes. And there are typically 2 ways of reconstructing the genomes. One is reference-based. For instance, the model species such as human already have a genome sequence as reference. And we can map all the sequence reads back to the reference genome to find the variations, mutations, to assemble a new genome. Or we could, without the reference templates, we can use the overlap information to assemble a complete new genome called de novo genome assembly. Now, from a computational point of view, the process mapping is somehow time-consuming, computationally intensive, because you have to compare each nucleotides to each other to find out the variations. And for de novo genome assembly, usually the state-of-art, the brain graph, stores all the k-mers and the adjacency information in the memory, and it's usually memory intensive. Of course, there are some new methods try to compress the brain graph, but still, it's a trade-off between speed and memory. So what in my case, my use case is to utilize powerful de.NBI cloud or any cloud resources to speed up the sequence mapping process and the de novo genome assembly process. And we harness the state-of-the-art parallel distributed computational engine called Spark. And the first tool I want to introduce is called SparkHit for sequence alignment. So here we have the de.NBI cloud, the infrastructure, where you have a lot of computational resources, CPUs, MEMS, network, so on and so forth. And use the OpenStack platform, we can virtualize all the resources into different so-called flavors. Instance with 32 cores, 60 gigabytes of RAM, and so on. But in your case, or in my case, I want to focus on the biological questions to solve the bioinformatic challenges. So I wouldn't spend too much time on building my own cluster. And here we have an in-house tool called BBGrid to easily set up a cluster, a SunGrid engine cluster that serves like the traditional servers that used in most of the in-house laboratories. But also for BBGrid, it's also able to set up a Spark cluster with just one single parameter specified to easily set up a Spark cluster. So what is a Spark? Spark in general is a distributed computational engine to facilitate your method implementation in a distributed fashion. It is basically extended MapReduce model that is based on Hadoop. It's very, let's say, commonly used in distributed combinations. And the advantage of Spark is that it enables users to cache the data into memories, and thus improve the performance of iterative algorithms. And the core for Spark framework is a distributed data abstraction or data structure called RDD, Resilient Distributed Data Sets. Now, Spark cluster is like a network that organized by a master node and several worker nodes. Of course, you can have more than one master node, 2 or 3. That's the cluster, basically the structure of the cluster. And then RDD is, from a computation point of view, is a Java object and RDD. And RDD is a data structure. So what RDD do is they automatically distribute your data, all the worker nodes. And all you have to focus is on implementing your own methods based on the RDD interface. For instance, here, you can program your own filter on map functions. And it will be automatically, this method will be automatically parallelized to all the partitions, a part of the data in the worker nodes, so that the distributed implementation becomes easier. And for a distributed engine, it usually should have fault-tolerant mechanisms. Because when you scale your cluster to more than 1,000, it's very likely that one of the cluster or one of the processes get disrupted or failed. So, as I have said, the RDD has a lot of interface and functions. You can implement your own methods. And this kind of functions or methods are called operations that have been categorized into 2 types, transformation and action. Transformation is an operation that applies on each partition, each part of your data independently. And the action is usually the case where you have to send back a result to the master node. That is usually not so parallelized. So this is steps of your, let's say a pipeline of your bioinformatic workflow. And this is, let's call it the lineage of your workflow. And once you have a failure happen to one of your worker nodes, the master node checks this incident and record your lineage and reschedule your failure process to the other working instance so that the complete process are intact and finished. So that's the framework I'm going to use because you don't want to write all the scheduling and stuff yourself. And for my case, I want to build a sequence alignment process. So basically, for sequence alignment, it's kind of easy to parallelize because it's a divide and conquer process. You just have to split your data into different chunks. Here, the data splitting process is managed by the RDD itself. What I have to do is I implement a mapping pipeline here. I'm not going to go into detail. This pipeline can be implemented as just a function of the RDD process. And this process will be automatically parallelized to each part of your worker nodes. But the mapping process is usually you map your sequencing data to part of the reference database or index. So what the Spark looks like is that we build the index just one of the nodes, on the master node or the driver nodes. And then we broadcast this one piece of reference index to each of the worker nodes. And then each worker nodes, each partition of this sequencing data, NGS data, map to the reference index and give you a feedback of the results. So this process is quite simple because it's divide and conquer. And so what we can do is that we can change of implementation to other tools, right? It's just basically a binary executables where we just have to send your data to an external resource, to an external executable binaries or a Docker containers. So that the Docker container manages the mapping process, the independent parallel process for you. So in this case, we could still here, each partition, you just have to apply another Docker function, or let's say a Piper function, where you download the Docker container, which is independent tools that can be executed on each worker node independently. Basically what here it does is you can replace my algorithm itself to other alignment tools such as BWA and time, so that our tools can be more robust. That's the first part of the mapping implementation. And the second part I want to present is a new algorithm or new tools that I developed to perform a parallel distributed genome assembly, reflexive. So when we talk about de novo genome assembly, usually the state-of-the-art algorithm or graph, it's called the Boy graph. It uses a k-mer. K-mer is a k length of nucleotides that extract from the genome. Use the k-mer, the overlap of the k-mers, to build a graph that can be traversed to extend the original genomes. Now, to distribute the Boy graph in a parallelized, in a cluster, in a distributed system, it's quite problematic because usually the Boy graph has a k-mer and as a node. A graph has a node as the k-mers and also have an edge that points to its adjacent nodes, k-mers, right? But in a distributed system, it is very likely that the two adjacent k-mers might not be in the same machine. So in addition to a node, an edge, usually you need a sort of pointer or an index to point to the physical location, which instance the adjacent k-mer is. So here, for instance, the Ray and Abyss implement this based on MPI-based distributed Boy graph. And now, for this kind of assemblers, the problem is it has a constant traffic. Start from the node 1, you might have to send to node 2, and then you have to send to node 3, and this kind of so on and so forth traffic that relies very heavily on the network. Thus, the assembler speed is quite slow. And what I developed is called reflexible distributed k-mer. Now I have to address reflexible means the k-mer will be reflected. Now, how do I define reflected? Let's take a look at here. We have a k-mer, ATGG. Reflected means, so this k-mer has two parts. You have a-1 suffix that is overlap with the next k-mer, the prefix of next k-mer. What I define reflect is that you swap the order of the prefix and suffix. You put the prefix to the end. Now, what you see here is that the two k-mers have the same prefix now. You know? As here, I did, here's an example. Now, first I reflect the first k-mer, A to T, and I sort the entire k-mer list. So now here in RDD, we only store the k-mers. We have no edges, no pointers, or whatsoever. Just think of it as a complete big list of all k-mers, and it's distributed into different machines. Now I reflect one of the k-mers and I sort them. Now in the big list, after the sorting, these two k-mers should be in the neighboring positions. And when you iterate it, you can easily extend this k-mer. And then you reflect again, but this time remember we still keep an n-1 because it's an overlap with the next k-mer. You reflect this k-mer, sort it again, and extend it. Right, this time it's extended to six nucleotides. And you reflect the 3 k-mers again, do it, extend it, you can get the original genomes. Now the question is, if you only reflect one of the k-mer every time you sort them through the entire cluster, it's going to be very time consuming. So what you can do is I randomly reflect half of the k-mers. So you have a big list of all the k-mers here, and you randomly reflect one of them. And you can see from here, every time you have a 2, 1, 2 means reflected, 1 means not reflected. 2 to 1 reflection, you will have after sorting, they will have adjacencies found in the big list. Which means that each k-mer, after random reflecting, each k-mer has 50% chance to connect with its neighboring k-mers. And after each iteration, there should be 25% or less. So in this case, only in the beginning, you have a big list of the k-mer lists, but afterwards, the computation complexity reduces dramatically. Here I give you another example which you might see more clear. This is an entire genome where you have all the k-mers listed there. And in the first iteration, I just randomly reflect them, right? 2, 1, 2, 1, 1, 2, 2, 1. And then after you sort in, the 2, 1 combination will be in the same location here. After the sorting is the red, blue combination. And then you just have to extend them after you get away. And then you do it again, but you do it completely randomly. Here you have another 2, 1, 2, 1, 2, 1 connection, and 2, 1 will be sorted together and extended. Yeah. Now let's look at it in a distributed way, because that's like a parallelization of just one machine. Here we have 3 worker nodes. So think of them as 3 memory slots. They don't know each other. Yeah? And you have a genome which has a lot of k-mers, all the k-mers. And what you do is you put all the k-mers into the memory, distribute it, and you do is randomly reflect all of them. Right? And then what you see is that the first 2 k-mers, they are after sorting, they are in the same place. And these 2 GTT also sorted in the same place. And you just have to extend them. And you do that again, you extend it, sort them, extend it. Extend, reflecting, sorting, extend it. Reflecting, sorting, extend it. And you get the results. Yeah, that's a genome assembly. So that's the two type of native implemented methods that I did on the Spark system. But again, as I said, you can also download Docker containers, executables, and other tools, different kind of tools, on the Spark cluster and to test this. So here I will present a use case that we did on Amazon Cloud, but it's the same methods that can be reproduced to the de.NBI Cloud, because we have some unspent credits on Amazon we want to use it. Together we aggregated 100 terabytes. I mean, just data is uncompressed, but the compressed size is 26 terabytes. Here let's start from the figure A. Figure A is a cluster setup process. We use BBGrid or other tools when you preferable to set up a cluster in the Amazon Cloud, which locates an island, because it's the cheapest. It's so to say, the cheapest server located at Amazon. And for BBGrid, let's say launch 100 nodes, computer nodes. It takes about 20 minutes to set up this cluster, where you just one line of command to type it in, and you wait 20 minutes for the cluster to come alive. And then what I did is we have the human microbial data sets that is stored in Oregon, USA. In total 8.6 terabytes, but the compression is 2.3 terabytes. So we download them, which took about 20 minutes, less than 15 minutes. And then we decompress them. Decompression also done in parallel by Spark in our frameworks. And we did some kind of fragment improvement, the sequence mapping, and also some profiling. And then we summarize the result and delete the database. just need to summarize the results. And then we again, without shutting down the server, we can download all the 3000 mice genome project located in Virginia, which is 15 terabytes of BEM files, aligned BEM files. We download them and use the MPIO, which is a binary tools to profile to detect the variants on the genomes. And then we delete the data, download all the 10,000 genome project, which are the FASTQ files, and we use BWA to map them to the reference genome. And so on and so forth. So in total, it took about 21 hours. So we have downloaded all the human microbial project, the sequencing data, and we map them to a selected 21 reference genome, microbial genomes. And here we have like seven representative of them. So what you get is the profile, the mapping profile, because we're using all the human microbiomes, different they are metagenomic data, this mix of all kinds of microbes. And if you map to all this reference genome, you can get kinds of ideas what the abundance, for instance, the abundance. Here is the identity of the mapping, and here is the normalized abundance of the mapping results, the profile of the species. You can see basically some kind of profile. For instance, here in the different human parts, like in stool, you have a very high abundance of E. coli. In the oral region, you have a high abundance of Trochilus mitis and stuff. Various kind of, depends on how you interpret it, this kind of thing. That's your specialties, I guess. Mine. Yeah, and that's the use case I have. I want to thank my supervisor, Alex Schober, and Jan Kruger, which is admin of de.NBI. You will meet afterwards. My study is funded by DD Graduate School, DFG funding, and also the infrastructure is provided by de.NBI.